is very happy to organize this webinar on the topic nanotechnology, evolution, and scope for next century on behalf of this National Science Day celebration of our college. So without much introduction, I invite our beloved Vice Principal Sir, Dr. Mansoor Ali Sir, to deliver the presidential address. So Mansoor Ali Sir, please.
Thank you, Mr. Tony.
Okay, thanks. Welcome.
Most respected resource person for the day, Dr. Swapna Nair, Head and Associate Professor of Department of Physics, Central University of Kasaragod, Head of the Department of Physics, Ms. Jinsi, Coordinator of this particular program, Mr. Tony, and other faculty colleagues, and my dearest students and fellow participants. It's indeed happy to be a part of this National Science Day celebration 2021, and we all know the theme for the National Science Day celebration for this year is Future STIs, Education Skills, right? And every year, February 28 for MES College Marambali is, we are organizing this National Science Day every year in a befitting manner. And we are, many departments, many science departments are coming with different program. And this year, this day, we are one, day ahead. Of course, this is the, weekend, Saturday, and tomorrow is, holiday. And maybe that's one of the reasons we are celebrating one day ahead. And National Science Day, we all know why the National Science Day is celebrated, so as to commemorate the Raman effect. That is one of the first Nobel prize for India come up with Raman. And indeed, it is exciting and good to see that our resource person has worked in Raman Research Institute, Bangalore, and that's a chair situated along with the Indian Institute of Science, and that's what my understanding. And anyway, the topic for the day is nanotechnology, and that is one of the thrust area. And we biologists, often collaborate. And recently, in MES, Marambali, we have done some, bio nano production, and that has been published in a, Q1 journal. And this is how, previously, the whole science are, in different rooms, and now started collaboration and cross-disciplinary or multidisciplinary kind of collaboration that will benefit more for the society. And that's what I learned from the resource person biodata also. She has been associated with the development of biosensors, and she is involved in materials for biology and medicine. And there is a scope for association for MES College Marabali also. And it's good to see that Department of Physics is organizing a webinar on thrust area nanotechnology and what is the evolution scope for the coming century. And we are having a very promising and dynamic and vibrant resource person to talk about nanotechnologies also. And now people are looking on top of you. And Robert Feynman, we all know the paraphrase. There are more room in the bottom. And this is indeed, we are in a situation, we are enjoying, the Mars Perseverance, landing there, and Ingenuity, the helicopter that want to roam around the Mars is, yet to start. And this is how the science is progressed. And this is one of the key indication that to search for life in other planets also. And that's where we stand now. And once again, I congratulate the Department of Physics for organizing and for celebrating National Science Day. And I am personally very happy to be a part of this National Science Day celebration. Once again, I welcome you, ma'am, for this webinar. And definitely, what I understand from you is that you are from Alva only, and someday and sometimes you may please visit our institute and interact with us. We will be blessed with your presence in our institute in offline mode also. Have a good day for all the participants, and I congratulate once again the Department of Physics. Thank you. Have a nice day. Thank you very much.
Okay, thank you, sir. Thank you very much for the presentation address. So, okay, now I would like to invite Dr. Swapna S. Nair, our resource person. person to deliver the talk. So before delivering the talk, I would like to tell some modesty about our resource person. So yeah, I know Sapna ma'am from personally from 2012 when I was a MPhil student at Central University of Kerala. So yeah, so there she was teaching us solid state physics, especially the condensed matter area of solid state physics. And what I personally experienced is that okay, so what we will tell is that okay, in our research scholars, there is usually a talk that yeah, the people are telling that okay, during your PhD time, if you are in trouble, then you will be in the right track. This is an ordinary talk in between the research scholars. So because you people feel that you will be in very trouble because of the supervisor's characters, because supervisor will never help you to those kinds of problems are there. So that you will definitely feel that okay, your PhD life is completely terrible, and so that yeah. So yeah, so then your PhD life will be in trouble, so that you will be in the right track. So this is the common talk. But what I personally experienced that okay, if you joined under Sopna ma'am as a project student or a PhD student, then you can see that you will never feel any trouble, because she is that much of student friendly, and that much of motivational inspirational researcher is some kind of person is that. So you will be in the right track, and also you will never feel any trouble. So this is my personal experience when I used to work with her, and these are the different feedback that I'm getting from her PhD students and project students, all those things. So that what I'm telling is that okay, if you want to work, so I'm telling to my students, if you want to work in the area of nanotechnology and you want to work in Kerala, so Sopna is the best option, because she is that much of student friendly and inspirational supervisor. So you will get. So this is one kind of modesty that I observed in her. So now, if we want to speak about her academic, so we know that yeah, she done her MSc and PhD from CUSAT, and then she worked as a postdoctoral fellow at the Institute of Nanomaterials and Nanodevices and Nanomodeling at Portugal. From 2008 to 2011. Then she again joined as a postdoctoral researcher at the CICUCO of University of Aveiro in January 2012. Then from 2012 onwards, she joined as a faculty member or assistant professor. Now she is an associate professor and head of the Central University of Kerala. Now she has guided almost 5 PhD students, and 3 of them have already submitted about the degree. Then now 2 of them has already submitted the thesis. Like that. And now she has many research articles and cited those articles more than 100,000 times in a very short span of time. And so anyhow, I don't want to if I want to speak much about her, there are a lot to speak, but I don't want to extend my introduction about her. So Sapna ma'am, I am inviting you to deliver the talk on the topic nanotechnology, evolution and scope for next century. Ma'am, so please.
Yeah. Is it audible? Is it audible?
Yes, ma'am. Yes, ma'am. It is audible.
Okay. A very warm good morning to one and all. Thank you, Tony, and thank you, sir, for the nice words. And I know there can be connection issues in between. If something happens in between, let me know by unmuting your mic. So. Today, actually, it is not the science day, but still only one day left for the science day. So I have to say, as Vice Principal Sir already mentioned about that, I have some association with Raman, because I worked in Raman Research Institute for a couple of years, means one to 2 years during my PhD term. I was working with Professor Raji Philip. And it was a nonlinear optical characterization of nanomaterials, and we could produce a device which was patented later. and the process also was patented for making an optical limiter made of ferrofluid. So Raman actually, when I'm talking about C.V. Raman, actually when he started Raman Research Institute, many times government tried to take the institute over and tried to merge it with Indian Institute of Science and all, but he never agreed for that. So it remains as Raman Research Institute. So it is giving an independent status for that particular institute. That was his aim, and so it is retained as such. And it is very close to the Indian Institute of Science, walkable distance. It is something like maybe less than 250 meters away from the main gate of the Indian Institute of Science. So, as you know, Science Day, it is being celebrated not as the birthday of, the date is selected not as the birthday of Raman. It's because we want science to be objective, not subjective. Anybody's birthday is not at all matter. What is invented is really matters, not who invented. So that is the difference between science and other disciplines, I have to say. If there is a scientific temper, other disciplines also has the same objectivity. Objectivity is there, we will be able to see that there is a scientific temper there. So that is why we never used to write I make or you make, okay, in a research article. So it is made. What is made is important than who made it or who discovered it. So with that message, I'm just starting my talk. Actually, I thought about many topics. Then I thought like nanotechnology is a topic where anybody can get something out of that. So I thought like I will start with the topic of nanotechnology. So the title gives many things, but may not be the content may not be giving that much because it is a very wide topic. Evolution and scope for next century means right from the beginning to end, we have lots to cover in nanotechnology because it's a very wide discipline. So as your vice principal sir told, so it is joining hands with all the other technologies. So it takes time to complete the discussion on everything. But I will try to cover some major aspects of this nanotechnology in the coming one forward. So as I told, 21st century just emerged with the balance of all these technologies, new technologies. We all know about these technologies: quantum technologies, artificial intelligence, robotics, then semiconductor technology, energy harvesters, energy storage, biotechnology. All these were there earlier also. Maybe a couple of years before also we have learned about this, we have heard about this. But now all these have gained momentum because people could understand the depth of application potential for all these. So semiconductor technology, it was there, but now you have to see IBM is going to just release their ultimate transistor, because ultimate in the sense, ultimate in lowest dimension, means its dimension is getting shrink to the maximum. So no more shrinking may be possible in the nearest possible, because unless we are going to just move away from the conventional technology, it is not possible for us to go beyond that or below that level, because they are going to shrink the transistors to its ultimate size. Then we need energy harvesters. So energy harvesters and energy storage, it is becoming very important, whether it is in nano range or in bulk range, because we want to use all these for many applications. So energy, because when population is increasing and the technology is enhancing, we need more energy harvesters and energy storage devices to supply quality life to the people, where everything is needed, and we need to search for novel materials which can deliver all these properties. So that is why all these are important. And then about robotics, artificial intelligence, quantum technologies, those are a little bit novel technologies. Still, people are trying to realize those technologies to its fullest extent. And then biotechnology. Biotechnology, actually, the basic biotechnology started much before, but now things are moved to such a range, like we can just edit any gene using CRISPR or siRNA or any other tools, and then we will be able to do. So Western medicine, they started talking about conceptualized individual medicines for individual patients based on the whatever be the biomarkers that are being developed. So in that case, biotechnology is also entering into a new stream, new a new era, and robotics. So all these are joining hands with nanotechnology, because nanotechnology has something to offer to all these technologies. That is why it is just being elaborated as a central technology, which is helping all other technologies, joining hands with all the new technologies. So everywhere in energy harvesters, in semiconductor technology is going to be nano from here onwards. The microelectronic mechanical systems are slowly giving way to the nano electromechanical systems. When I will go to that particular section, I will elaborate how we change from the bulk systems to the nano. Then so energy is again, it is being helped by nanotechnology, nanoporous materials for energy storage. Lots of such applications are there for nanotechnology and energy storage. Biotechnology. In biotechnology, lots and lots of application. I will start with biotechnology application because it is an era where we all are concerned about our health era. So that is why I'm going to start with the application of nanotechnology in biosciences first. So uh, I'm not going to give a wide discussion on what is nano and on those aspects, because you all know about nano. Nano is nothing but 1 billionth of a meter. So nano world, I just have shown some examples of the nano world where this is a graphene sheet, and this is a buckyball structure, a soccer ball structure. This is a carbon nanotube. Okay, pointer is not. Okay, this is a carbon nanotube, and this is a buckyball-like structure. This is a graphene sheet, and we will be able to see superlattices and then DNA. So I have to say these are man-made materials. All these are man-made materials. Somewhere there, and somewhere there early, but most of them are man-made, tailor-made materials, because they were materials, but we changed their properties and made it like this. It is a carbon nanosheet where carbon atoms are arranged hexagonally. It is called graphene. It is a single layer of carbon nanosheet, and we will have multiple layers. Maybe sometimes we will be able to exfoliate a single layer out of that. So this, through the exfoliation, we made it different because it was graphite earlier. And this is a quantum dot solution, so it just changes its color with respect to its size. So it's a wonderful property of this quantum dots, and we will be able to see some. We know that it's a pic of double helix DNA. So we will be able to see that, okay, now the materials were there in nature much before, but uh, as your teacher rightly said, so it is like we didn't have opportunity to hop into the nanostructure before the invention of electron microscopes. So that is why people were not that fascinated about the size before that, because they were not able to, they can imagine, but they were not able to see the structures. When we don't have tools, we are not that fortunate enough to see the structures, and we will not be able to study the structure. But once the electron microscopes were invented, after that, the whatever be the speculations or principles, concepts, everything in scientists' mind, they could prove it easily. That is why, in most cases, sophisticated equipments are needed to probe into the structure and properties of nanomaterials, because they are ultimately very, very small. So we will go to the next slide, in which I have shown some specific structures that can be called as nano. I will just explain those. This is a nano rod and a nano wire. So what is the basic difference between a nano rod and a nano wire? It is the aspect ratio. If the aspect ratio means length to the cross section. So if it is very small, just like 1 to 3 or 1 to 4, then it is a nano rod, because it is a rod. It is a thicker one as compared to this one. This is thinner in the sense compared to the length. Okay, so length is very big, and the cross section as compared to the length is very, very small in nano wires. But in nano rods, it is aspect ratio is coming in and around 1 to 4. That is the basic difference between nano rod and nano wire. Otherwise, everything is same, because only length matters. And this is a tube. Tube is again a one-dimensional nanostructure, but a nanotube has hollow in structure. So obviously, it is clear from the nomenclature itself. It's a nanotube. And this is a nano-whisker because it has got whisker-like appearance, and this is a quantum dot solution. So try to see the color variation. It's of the same material. Earlier, people believed that the color is a property of a material. But now, coming to the nano range, I have to say there is nothing like a property for a particular material. Now, everything is tunable. So I can make my material emit in blue. I can make my material emit in UV. I can make the material to emit in IR. So everything is tunable. I can just incorporate defect states. I can just engineer with the defects. I can add some dopants. So everything is possible. So, and then you have to understand there is a fundamental property which is, I don't know how many of you are physics students. Physics and chemistry students were knowing about quantum confinement. So because of the quantum confinement in nanomaterials, the band gap is shifted to the higher energy range. So even a metal can turn into semiconductor, and a semiconductor can turn into insulator in nano. So that is why the material property is not more the material property. It is the size property, size and shape properties. Completely the properties are determined by the size and shape rather than the material. So conventionally, if I speak about gold, I know that gold has some lustrous appearances. It is close to some yellowish color. So, but in nano, it is not like that, because this solution itself shows in nano, gold can be like this. It is because of surface plasmon resonance. The details and all I am not going to explain it here, but you will be able to see from this structure. Okay, this graph shows the size-dependent photoluminescence property. So this is luminescence. The y-axis is luminescence, and the x-axis is lambda, which is the wavelength. So you will be able to say that we will be able to tune the fluorescence in such a way that we will be able to make the material emit. That is called tailorability. Okay, if I want my material to emit, if I want to make a laser or a luminescent devices or an LED or whatever be the device, if I want my material to emit at 430, I can make that material by tuning the size and shape of the material. That is the wonderful thing that we will be able to. And I may not, if I'm an expert in chemical synthesis techniques, I may not need sophisticated equipments. I can make it in seconds. So just with a pH alteration or a temperature change or a play with some surfactant chemistry. So some energy balances, all these, if I can do within seconds, I will be able to make it with a simple test tube. I will be able to make the material in different size ranges, which can emit differently, the same material with different luminescence. So that is what is shown in this. So luminescence, the peak is being shifted from this 450 range to the 650 range. Why tunability? Means the same material can emit in blue, and it can emit in green, it can emit in greenish blue, it can emit in yellow, so it can emit in red, or maybe in IR range or UV range. That much tailorability is there for a nanomaterial if I can play with the size. So that is making the application multifold, because I can even make a semiconductor as well as insulator from the same material. I can make metal, insulator, and semiconductor, everything in the same material by playing with this size, shape, and the surface defects. So that is why this nano world is really interesting, because we can use these nanomaterials for a variety of applications, like starting from the luminescent devices, the IV photovoltaic cells. So because of the tailorability of the bandage, it can absorb anything, it can emit anything. So it is possible for us to tailor the properties through the synthesis techniques, simply by altering the synthesis techniques. So that is why it is very important. Okay. So now, as I told, it is an era where we all are troubled with lots of things. Our lifestyle itself has changed a lot, right, after the COVID outbreak. So we are slowly trying to adjust with the new lifestyle. So I have to say, if I'm just forgetting about the role of nanotechnology in bio, it is actually worthless. So first I'm starting their applications in biotechnology, because biotechnology, as well as a novel field, as I told, after the invention, okay, after the advancement that is being happening in the field of genetic engineering, I have to say nanotechnology and biotechnology. Now they're joining hands, and nano serves as a platform for most of these biotechnology applications nowadays, whether it is in agriculture, whether it is in food technology, whether it is in real genetic engineering, whether it is in nanomedicine. It has good role, it has good potential to offer, so that the biotechnology, whatever be the tools that are being used for the biotechnology, can get another fold advancement by incorporating the nano tools. So nanodiagnostics is one among the base application of nanotechnology, because we know prevention is better than cure. So if I can understand some signatures that are there much early, then we will be able to cure, or maybe we will be able to prevent that occurrence of that particular disease. So nanodiagnostics, diagnostics using nano tools, that is what nanodiagnostics, simple. So that serves as a platform for early and accurate diagnostics, which is very important in treatment, or even to prevent whatever be the issues that are going to happen to us. So, for which the role of nanotechnology is to develop high sensors, very small ones, which can even go inside our body and then come back with the whatever be the information that is needed. So that is why it is very important. And then imaging, so targeted imaging. So it can go, the nano tool can just go through the body and then go to some desired location and give images of that particular location. So that is how it is very important. Okay, it is giving very important ideas about the inside of the structure, body, and even if it is cellular model, we will be getting information about small okay, just like inside cells there are a lot of vesicles and all. We will be able to target that into a very small desired location, and we will be able to image that particular location, whether there is any damage or any signature of any particular diseases. So that is why nanodiagnostics, there is a wide scope for nanotechnology to offer, so that we will be able to make ourselves healthy, long living, and that is the scope for that particular technology. And the second one is targeted drug delivery. Targeted drug delivery, so it is, by the name itself, it is clear that we are going to target the drug to the desired location. Just like the way I was talking about the sensors or the imaging tool, drug also can be delivered to the desired location. That will really lower the impact, because most of the drugs has very bad side effects often, because we have to take the drugs to relieve the whatever be the issues, but we have to see that they may be so if I can carefully target the medicine to the desired location only, then the off-target effects can get reduced. So in that cases, the other cells or other healthy cells won't be affected much, because of the targeted drug delivery aspect. So there also, nano can serve as a platform for targeted drug delivery. So in regenerative medicine, again, stimulated repaires, then some other things like genetic editing. Nowadays, nano serves as a platform for gene editing tools like CRISPR as well as siRNA. So lots of applications are there in nanobiotechnology. And now I'll come to one of those where physics students may find much interest. It is the hyperthermia. Hyperthermia, from the word itself, it is clear that hyperthermia, high temperature, it is inducing high temperature and then killing the unwanted cells. So wherever we want to kill the cells, we will be able to use this particular use. Magnetic nanoparticles are widely used for this particular process, because you know about magnetic nanoparticles and the hysteresis. Okay, so hysteresis, if I have to say, let me see, it's not possible to, okay, because Zoom only has those pen option available here. So I will not be able to show the pen option, but it's okay. I will try to explain without a pen. So you know about magnetic hysteresis and magnetic relaxation. So in each hysteresis, means each cycle of magnetization, there is an energy loss. So you have to see that energy loss can be made into, so it is a loss, energy loss. We don't want any loss, but here that loss itself can be made advantageous. Okay, how? Because in each cycle of magnetization, there is an energy loss which will appear as heat. So if I can enhance the number of such repetition, means if I am giving a kilohertz magnetic field, AC magnetic field, then it is possible for me to make the whatever be um, uh, the energy loss. The heat generated will give us something like high temperature range, just like 5 degrees to 6 degree rise in the temperature if I am giving a kilohertz frequency of magnetic field. So that will selectively kill the undesired portion, just like a cancer cell, because I can really target the magnetic nanoparticle towards the tumor location. This shows. So this is a biocompatible magnetic nanoparticle. Generally, we use superparamagnetic iron oxide for this particular process because it is very less as far as toxicity is concerned. Its toxicity is very, very less as compared to other systems. So biocompatible magnetic nanoparticle is injected and it is going to the stream, and there are specific surface modifications so that it can go to the tumor cell selectively. So just like if it's a breast cancer cell, it has a good folate receptor. So we caught the material with folic acid. Just like that, based on the particular biomarker where we want to target, then based on that we will be able to modify the surface chemistry of nanomaterial so that this nanomaterial will selectively go and stick to that particular location only. It will not, so that will really lower the hazardous off-target effects because it is going to the targeted location only and it is not going to harm other cells. So that is very important as far as medicine is concerned. So it can go to the tumor directly. Oh, sorry, something is happening whenever I'm touching. I'm trying to take the tool. Sorry. So this is the tumor, and this will go and selectively go to the tumor. So that is what is shown here, and the particle is here only, and I can give the field. This is a field. It shows the field. This shows the field. So this is the field. So we are giving the magnetic field so that maybe after some cycles of magnetization, this will finally give the temperature rise which is required to kill that particular tumor cells. So this is wonderful as far as a cancer therapy is concerned because it reduces the off-target effects. And now it is possible for you to see how nanoparticles are taken from the cells. So once the nanoparticle is properly surface modified, it will go and it will go to the side of the cell. So this is a cell. This bigger one is a cell. So cell surface. Those who are not from the biology background, I'm explaining all these. Otherwise, they will feel others, those who are coming from biology field, they will get bored, I know. But these are essential things that may not be known to the physics and chemistry students, so I'm explaining this. This is a cell surface, and this nanoparticle goes, and generally nanoparticles are uptaken through this particular process of evagination that is called endocytosis. So once it appears here, the cell feels the pressure and it evagenates, and the nanoparticles are engulfed inside. It is called endocytosis, and these nanoparticles are taken up to the desired location by properly modifying the surface. That is why the surface modification is shown here. Properly, we have to modify the surface so as to make them appear in that particular location only. Okay, now I can apply the magnetic field and do whatever be the repair that is needed. So here it is used for magnetic resonance imaging. So for that also we nowadays use ferrofluids or ultrafine nanoparticles and magnetic nanoparticles that are suspended. So generally earlier days people were using gadolinium particles which were found toxic for the liver and kidney, and because of that nowadays people are focusing on ferrofluids. Actually my PhD topic was on ferrofluids. So we have done some experiments in that aspect. So for magnetic resonance imaging, nowadays ferrofluid is a material that is widely used for enhancing the contrast of magnetic resonance imaging. I'll be able to see this particular paper which is published recently. So innovative magnetic nanoparticles for PET. It's not our paper, it's a paper published paper by this particular group, Professor Malat Nadine and his group. and innovative magnetic nanoparticles for PET and MRI bimodal imaging. So they are using the nanoparticles for bimodal imaging. So PET as well as MRI is overlapped. The signal is getting overlaid, and then it is possible for us to get more information from this. And now try to see here the application is doubled, because this magnetic fluid is actually so magnetic fluid, as I told, it is very good tool to do the hyperthermia. But here the cancer drug is also added to the magnetic nanofluids, and it is being focused to that particular location, and this is that location, and magnetic particles because of this field, the field is shown here, the magnet is shown here. Because of the AC modulating fields, the magnetic nanoparticles temperature rise will be there, hypothermia will be there, temperature rise up to the range of 5 degrees Celsius will be there, and after the drug also will be released. So chemotherapy as well as hypothermia can be done using the same system of nanoparticles. That is why it is very important. These nanoparticles have lots of advantages, because it can serve in multifold way. It can carry the drug, it can deliver the drug, it can focus onto the desired location. So targeting is there, drug delivery is there, then apart from that, hyperthermia also is there. Using the same set of nanoparticles, we need not use many platforms for delivering all these stuffs. So very important. And I have to say, the things that are coming in sub 5 nanometer, it is very difficult to synthesize using wet chemistry routes, but some experts in wet chemistry routes can really make those type of materials by playing with the parameters, wet chemistry parameters, and we will be able to make sub 5 nanometer particles. Okay, in my lab, most of my research scholars are able to make sub 5 nanometer particles through simple wet chemistry routes, and these sub 5 nanometer particles have a lot of advantages, you have to say, because they can be expelled through urine, because it will just pass through the renal system, and we can expel that through the urine. That will reduce the toxicity, and liver and kidney loading can be reduced a lot. So we'll go to the next slide, where imaging and the antibody release, DNA. So genetic material and antibodies can be sent to the desired location, delivered to the desired location using these particular nano tools. So gold nanoparticles are widely being used for this particular processes, but nowadays other biocompatible materials are being developed, because gold is extremely costly. I don't want to use gold because it is very costly, but I have to make a material which is biocompatible. That is why you are needed, your brain is needed, because in nanotechnology, making nanomaterials of different kinds is not that of much interest, I have to say. But we have to make some specific materials which is having multifold dimensional application potential. So there lies the importance. So gold is a wonderful material, very low toxicity, it can deliver everything, but it's costly. I don't want that much costly material. So if I want to make another material which has the same level of properties, maybe a little bit trade-off, then still it is okay, because gold is much costlier. So I can make a substitute for these. People will accept that. Fluorescent carbon nanoparticles emerge as another material for these particular applications. So you see, you will be able to make this gold nanoparticle with that is why it is called the multi-dimensional potential. You will be able to say DNA is there, antibody is there, and then polymer also is there. This polymer will help to tailor the bandage and make the material biocompatible a little bit more. And then DNA is there. It is for delivering DNA to some specific location, and the antibody also is there. So it is a multi-dimensioned delivery. Okay, so that is why it is very important. See the case here. You will be able to say the case. The mice is there. There is a tumor here, and we are trying to use this nanotechnology for sensing, imaging, drug delivery, and therapy simultaneously. Okay, so we are using the same same system as a sensor, same system as a therapy agent, same system as a drug delivery agent, and same system for imaging. So that is why it is very, very important, because these are having, just like gold nanoparticles, copper nanoparticles, all these are having tunable surface plasmon resonance, which will make them useful for imaging applications. So imaging is covered now. Now, it has got absorption band in IR range, so that localized heat generation can be there. So it can be used for hypothermia. That is called therapy. So we can use it for therapy. And then using other surface modification, we will be able to deliver drug delivery. So the same can be used for drug delivery. And if the same material has sensing properties, some specific sensing that will give us some signature about some diseases, then it is wonderful. Sensor is there, imaging is there, drug delivery is there, therapy is there, a complete package is there. And nanomaterial is giving this complete package. That kind of material is the need of the hour. So, here also gold nanoparticles, it has got all these diagnostics potential, drug delivery potential, therapy, imaging, all this potential is there. And it can deliver gene, protein, drug, and what else? Anything. So nowadays people started using this as a platform for delivering CRISPR. So earlier there was something like a picture, a video that is released by BBC. BBC often comes with this 5 year plus technologies, just like the technology that is going to come in 5 years. So that they will just envisage and make us videos. In that video, it was given like a nano robot is going over there, here, and delivering all this in desired location and coming out changing anything, and at certain particular portions, they are creating some changes in the whatever be the if there are some issues in the system, it can correct, repair, and then come back. So do some genetic editing and then come back. If it is something like a disease that is going to happen, we will be able to predict that and then do the corrections and make the person healthy. He will be able to come up as a healthy person. So, and then photothermal therapy, radiation therapy, all these we will be able to use this kind of nanoparticles. And gold is a wonderful material, but the cost is the issue. And these are some other examples. I already have explained all these things. Delivery of genetic material here. This is an siRNA. Here we have a project with CPCRA Kasaragod in treating a particular disease by genetic editing. So siRNA is being delivered through a nanomaterial. So that is how nanotechnology can help in that particular idea. Okay. Complementary research. We know something, we are using our whatever be the knowledge that is in our hand. Biologists may have some ideas, and we will think together so that if I can help in in achieving their goals, then it is a new invention. So that is how your vice principals are rightly said. The era of focused research is no more there. Focusing is required. And we need to develop our core area, but we have to see whether the development that is going to come in core area can help somebody else, can help the humanity in general. So because that is why the health is so important for everybody, right? So if we can contribute whatever be the knowledge that is gathered by us to the health of the humankind or animal kind or plant kind, that will be wonderful. So that is why applications of these genetic engineering using nano tools, if it is available, if I can make such a material which can deliver the siRNA or CRISPR to do the gene editing, and then it will be wonderful. So that is why it is all important. Okay, so then I have to talk about the research work that is being conducted in our group. I have two students, those who are working in this particular area, Neeli Chandran and Maniganda, and they are joining hands with my collaborator, Dr. Rajendra. He's a Harvard alumni, and he's there as the Dean of School of Medicine and Public Health in Central University of Kerala. We are joining hands in all these aspects: nanodrugs, drug targeting, diagnosis, sensors, imaging, all these aspects. So two of my students, I just have made specifically their aims are to develop nanodrugs and drug targeting, diagnosis, nanosensors, biosensors, and nanomaterials for bioimaging, etc. So these are the areas where we are focusing, and we are doing some research work with some other institutions like in CPCRA and some other applications collaborating with Cochin University of Science and Technology. So it is a combined effort. Everybody. So my potential collaborator is Dr. Rajendra from Central University of Kerala. So he, for the nanodrugs, the drug targeting, diagnosis, sensors, imaging, etc., we are strongly collaborating with his group. The virologist, so we are trying to use whatever be the expertise that is in our hand for viral vector delivery, viral drug delivery, etc. also. So these are some ideas where it is envisaged as nanovectors for therapeutic agents. You will be able to say antibodies and their conjugates and viral vectors. So we will be able to use these particular things for treating the whatever be the diseases, viral diseases, and nanoparticles for imaging. It is another thing which is developed in our lab, copper with glutathione. This work is published in ACS Applied Biomaterials recently, and copper is linked with glutathione. So Neeli Chandran and Prajit Janardhanan. So as I told, I have a bio counterpart. So a student from the biology group and my group joined hands together to make this work. So it is always important to find proper collaborations and then do the things in a perfect way. So copper, as I told, it can turn as a substitute for gold in all this, because we will be able to tune the fluorescence, the surface plasmon resonance, everything in copper also. There were reports earlier also, and we could make it work for this particular paper. And this can image really with copper and glutathione, the complex finally first emit in blue, and later it started giving a fluorescence in red. So we use that for, so this is another nanoparticle system where we could use the zinc sulfide nanoparticles for imaging. So zinc sulfide, my student Mr. Manikanta is working with these zinc sulfide nanoparticles. Anyways, we are making some 5 nanometer particles. It is very difficult to make, but once it is optimized, it is well and good, because it can turn very, very biocompatible with very low toxicity levels. So zinc sulfide, out of the several systems that we have tried in our lab, we could find that zinc sulfide and copper, it is highly biocompatible. At this particular size range, I have to say, there is a variation when the size is enhancing. So you'll be able to see the treatment of these tumor cells here. So it is it's not a work from our group. It's for your knowledge I'm showing. So the tumor is there, and it is imaged, and then the same system is delivering drugs slowly. Okay, after several hours, it is again imaged, and we will be able to see that after several hours later, 36 hours later, the tumor disappears. So it is able to do that work. So this is a sensing work developed in our lab. So this copper glutathione, this shows that the fluorescence of the copper glutathione, and it has got high potential to be so it can sense the pH, as well as it can sense hypoxia and temperature. So very good platform, right? So cellular thermometer. So it can measure the temperature of the cells, inside the cells, and it can serve as a hypoxia sensor. Hypoxia, as you know, sometimes hypoxia is an early signature of heart attack. If it can be predicted much earlier, it has got high level of clinical importance. So all these are important. We are using our knowledge for the benefit of whatever be the human society or animal society or living things in general. So that is where we can contribute a lot. So this is a sensor that is developed in our lab copper with glutathione, and how it can be made and all it is showing in this particular uh, figure, and it is indicative of oxidative stress. So that means cancer was a sensor for hypoxia. And there are reports of carbon nanotubes for peptide delivery. By properly modifying the surface, we can load peptides, we can load siRNA, we can load DNA, we can load drugs, everything, and we can deliver that in the specific sites, and then take away the carrier. That is our nanoparticle. So that is why this is a wonderful thing. Just like we are just sending a robot to the body and then deliver everything in desired location, come back, and then forget about that. So that is a wonderful thing. That is why nanotechnology is important in health and medicine a lot. So drug delivery, it shows how drugs can be delivered, and now drug formulation. So the other thing was drug delivery. Now drug formulation also has a role. We will be able to make some specific nanoparticles which are completely biocompatible, and then use that as a drug along with a potential drug. So it is called nanodrug formulation. So we are making a composites of nanomaterial and this drug, and then deliver that to the desired location. And sometimes this nanoparticle will help us to image also, because most of these nanoparticles, if properly tailored, can emit in a particular wavelength. So we will be aiming inside, and we will be able to target, okay, live monitoring of the whatever be the things that are happening inside. That is possible because nano serves as a platform for imaging because of its fluorescence. Okay, most of the nanomaterials are fluorescent, as I told. I showed you, right, some of the nanoparticles which are developed in my lab. So this is a copper glutathione. This is a copper glutathione complex. It shows the fluorescence in red. So lots of such materials are there which can emit in blue, which can emit in green, or whatever be the emission that we want. We can make nanoparticles accordingly so that it will give that particular fluorescence, and we can use that for imaging applications. So because sometimes the cell inside cell we will stain some area with some other dye, and if I'm using the same, it's using a nanomaterial which will emit in the same wavelength, it is useless. Just to play with the grain size, surface chemistry, and all, and make them emit in particular wavelength. Then that is fine. We will be able to co-localize and take the images where nanoparticle is going and what is happening inside. So through live monitoring, we will be able to do the imaging. So this is another work that is done in our lab. So nowadays we all are worried about viral diseases, right? So this is a drug that is developed for dengue virus inhibition with lauric acid and zinc sulfide. So this result is published already, and then here we have to see that by we earlier days we thought like nanomaterial can increase the toxicity, but see this particular result, nanomaterials actually really reduce the toxicity. So pure lauric acid, it is like the LD50 is 1.25 micromolar, IC50 is 2.5 micromolar. LD50 is the dose which is required. IC50 is 2.5 micromolar. So LD50 and IC50 may not be known to the physics and chemistry students. So I'm explaining that in terms which is known to them. So 2.0 micromolar concentration, if we are giving, then we will be able to inhibit. Okay, and then 1.25, this is something like just reduced. Okay, 5. Yeah, 1.25 micromolar. It's a LD50, so it's a 50% depth. So it is something like the therapeutic index is not that very good if we are getting only double of the value. So if I am taking a paracetamol 500 milligram, if my headache will go only after using 2 tablets, means 1000 milligram, and after 2000 milligram it is highly toxic. Okay, in that case, I will not be able to use that particular thing because toxicity levels is very high. So we have to make the therapeutic index a little bit higher so that we'll be able to use that. So in Sardana's lauric acid nanodrug formulation, it shows that the therapeutic index is much higher. I think LD50 and IC50 is written in opposite way. Something happened. Okay. Anyway, we need an inhibitory concentration which is much less, and we need the lethal dose where 50 percentage will be killed. That is very high. So in that case, we will get a very good what you call drug. So here it shows that the combination of Sardana's drug with this whatever be the lauric acid, that really reduce the toxicity a lot. Another order. So maybe from 2 means it was the index was 2, and later it got changed to 50 to 100 range. Means 2 order enhancement is there. That is why it can be understood in the same terms of safety index is much higher in nanodrug formulation. Why it is happening? It is because of the slow delivery of this particular drug. Again, another drug formulation which is made in our lab in collaboration with the group, as I mentioned, with Dr. Rajendra and his group. And this is Sardana's carboplatin nanodrug for treating cancer. So the carboplatin is a drug that is widely used in cancer treatment. Here also, the in pure Sardana's and nanodrug combination, as compared to the pure carboplatin, pure drug combination, the value is much whatever be the LD50 value is much improved in nanodrug formulation. So another thing, taking advantage of retention. So now I have to say another thing. L'Oreal now came up with a set of beauty products. If you search on the web, you will be able to say nano enhanced beauty products. So why it is important? Because these nanoparticles, if it is part of this particular, whatever be the cosmetic agent like glutathione or some other cosmetic agent, if it is coated on top of the nanomaterial, this nanomaterial is these nanoparticles are very small. It can pass through the membranes, just deliver the things in desired location. If the fatty acid molecules are coated just like that, only through hair follicles it can go. So this nano serves as a carrier because it has got a different uptake policy. So a cell will take up through the invagination and endocytosis, and we will be able to deliver the material there inside. And this particular thing shows how it can be even if it is not properly surface modified, it will go and the portion where we have the leaky vasculature, just like a tumor location, it will automatically detect, because leaky vasculature only allows the accumulation. So at this particular region, the nanoparticles will be accumulated. If this nanoparticle is coated with drug, that also will be accumulated. So that is why it is very important. Okay, tumor tissue, it can directly go to the tumor tissue range. So factors affecting uptake, size, shape, dependence, all these we have published in another journal. So barriers to nanovectors delivery. So there are endothelial and epithelial barrier and reticuloendothelial system. Then there can have enzymatic degradation once it is going inside our body, we don't know. And many other things like cell membrane, nuclear membrane. So pumps are there, calcium pump, molecular pumps, a lot of pumps are there. So where it will go, sometimes it can be expelled outside. So without doing whatever be the intended work. So these are some drawbacks and challenges. Scientists are trying to just overcome these challenges through different routes. Because a smaller nanoparticle can be a solution for most of these initial, whatever be the barriers, while some other modifications are really required to bypass the ionic and molecular pump and expulsion through them. And this shows the background chip that is developed for understanding the early signatures of the prostate-specific antigen. Means chances of prostate cancer, development of prostate cancer. So here it shows that 3 cantilevers are coated with 3 different antibodies. device. Okay, so we will be able to say PSA. The left cantilever bends as PSA binds to the anti-PSA body, because this only has because the cantilevers are coated with antibodies, right? So if PSA antigen is there, that will go and selectively bind in that particular location. So we will be able to say that this particular this particular cantilever is bent because weight is more. You have to understand it is in a very small weight fraction, but still we will be able to understand the bending. Even if it is a very small weight change, we will be able to identify using laser system. So that is how we will be able to understand the presence. That is how sensors are being developed. Okay, very small mass changes can be understood, and we will be able to sense that early signatures of prostate cancer possibility. Prostate cancer possibility. So this particular Biocom chip shows how we will be able to predict that. So this even this cantilever itself is not required, and a nanotube which is properly coated also can be used for this particular process. A graphene sheet properly modified can also serve as a platform. It's all ideas. It's all ideas. If you can imagine something, you will be able to make it, and that is a wonderful thing. So if you can imagine only, try to imagine at least, so that others can get benefit of your imagination. So that is how nanotechnology is growing. So sometimes small children can give some revolutionize whatever be the happenings that are coming in nanoscience. So now I'm stopping my discussion on bio because it's almost a long time, and I have to go to my core topic, which is semiconductor technology. So you all know that it is an era of Internet of Things. So networking, so that is the important thing that is going to happen in our life soon. So now I know that it is networked already, because maybe during my master, because you are master students mostly and graduate students. When I was in graduation, I didn't have a cell phone. And cell phones were not yet in market. Okay, when I started my masters in the year 2000, yeah, no, 1999, it was not there. Yeah, in 2000, I got my first Nokia 3300 phone in my hand, I think, if my memory is correct. So that was not a smartphone. Okay, so from there, how much change had happened, how much revolutions have happened, and now it is possible for, even not now, maybe in 10 years before itself, it was possible for us to talk to our parents. Even if we are sitting in US, we will be able to have video chats. Now it is no more something like a curious thing. So everybody can use that, and everybody is connected. I don't think there is even one student left in class who don't have access to the smart medias. Laptops, iPads, then whatever be the mobile phones, all are smart devices which can be interconnected soon. So